So let's move on with the program. So it's a great pleasure to introduce Susana Huelga and from University of Ulm. And Susana will be updating us on the coherent dynamics in pigment protein complexes. So Susana, please.
Yeah, thanks, Jyrki. Yeah, I thought that despite pigment protein complexes are a bit of an exotic scenario, but as I've talked about these issues in previous editions, so I think it maybe is a good idea that I provide you with an update of how things have been evolving, yeah, and also as an illustration of how the methods and the techniques we are developing in the context of open system dynamics may actually be helpful in this arena that covers is more the territory of chemical physics traditionally. So some of you know some of these results, especially the initial part, so apologize for the repetition, but I believe there is other people that maybe they hear it for the first time. So I'll try to present sort of self-contained view, a little bit focused, not to say entirely focused on our own results, but let's say in the sake of completeness, I think is easier that way. So fixing the scenario, we are talking about our systems of interest are going to be hybrid systems, as you will see in a second, that are actually membrane proteins that are hosted in the cellular membrane of different organisms, and that have a transducing role. So upon irradiation, they convey charge or energy in such a manner that the quantum yield is very, very high, despite they operate in very noisy conditions. So there is a large phenomenology involving the primary state of photosynthesis of the radical pair formation that is thought to underpin the magnetic compass that birds use to navigate, possibly also involving primary state of vision related to retinal isomerization. And given that the scenario where we had the largest body of experimental data available is in photosynthesis, I will concentrate the discussion and use the experimental results that concern this type of complexes. Right. Recently, there are other investigations of possible coherent effects outside this paradigm of ultra-fast processes that concern allosteric regulation of chiral-induced spin selectivity, but that's something that I will not touch upon at all. So the first thing I want to do is very briefly to describe you the type of complexes we are talking about, and specifically address the question of what is this wording of long-lived coherence mean exactly, and why these complexes abandoned the chemical physics territory to actually attract the interest of the open system community. And then discuss the type of models that we and many other groups have put forward to try to understand both the dynamics and the spectral response of these open quantum systems. As you will see, anticipating already the results, is the difficulty of putting these two things together, what is preventing a full understanding on what is going on, or what the possible functionality of these complexes is. Right. So the James Cummings model of quantum bio is in fact the Fena-Matthew-Olson complex. That is a molecular aggregate that appears in trimeric form and is quite simple for the biochemical point of view, given that it only involves 8 highly absorbing molecules, 8 bacteriochlorophylls. So we are dealing with systems that involve electronic and vibrational degrees of freedom. You will see that our consideration of what is our system of interest and what should we think the environment made of is going to be changing along the talk. But as far as the electronic degrees of freedoms are concerned, in the moment that these complexes irradiated and electronic transitions happen, then dipolar interaction between chromophores will kick on. And this can be described by a typical exchange interaction of this form. So the first contribution to the Hamiltonian dynamics is just the side energies of the pigments, and then excitations can migrate across this network via a dipolar interaction of certain strength. Take into account that by means of this coupling, then electronic states that corresponds to a delocalized excitation will form, and I will call those excitonic states. So they are really simply the eigenstates of this electronic Hamiltonian, right? And then we have a plethora of vibrations around. They come both from intramolecular modes of the chromophores, right? And we also have vibrations that correspond to the protein motion itself. So this cage that is holding the chromophores into place is far from static, and is also the source of the slow protein motion, will have some contribution to the overall spectral density from them, and as I said before, from the intramolecular modes themselves. So if we think about a distinction between electronic degrees of freedom as our system of interest, and we put all the vibrations as our, let's say, environment, we will see that the effect of vibrations is going to vary from, in the first instance, yes, really providing the reservoir that would allow the dissipative dynamics of excitonic states. And then they will evolve into becoming part of the system and actually provide us with the vibronic model that we believe can account for several relevant features. Right, so now if you look at this Hamiltonian and you would like to start playing with it, you realize that I need some entries for the side energies, and I need in between pigments is the strength of the electronic interaction, right? So how are those numbers obtained? Well, this is a mixed technique, let's say, that combines from first principle calculations, right, to information that comes, should we have it, from the X-ray crystallography itself, concerning the orientation and the distance between pigments, but mainly comes from fitting a very, very large body of spectroscopy data. So for instance, here I have different figures that correspond to absorption, to some forms of dichroism. And the point is that making fittings with some form of the spectrum of the vibrational fluctuations, right, that can be measured, or also a combination of measured and molecular dynamics results, then we can provide a kick-on. Here I have a list of a possible set of side energies, right? And let's say more and more consistency with new experimental data allows to refine the considered values. So you will actually see that this is key to understand what happens. Moreover, besides the information we have from linear spectroscopy, there is also a large body of experimental data that come now from non-linear spectroscopy. So that's it. Instead of having just a single irradiation, now what we do is interrogate the sample by means of sequences of laser pulses. 2 would be a typical PAM probe. More sophisticated, you can have 3 ultra-short laser pulses whose time difference can be controlled, right, in such a way that we can actually end up with a 2D map, or a correlation map, if you want, between the excitation and emission frequencies. We don't need to enter into the details, which are actually quite complicated, but I just emphasize that our body of parameters needs to be consistent with whatever. We need a single set that is able to account for all the different forms of spectroscopy. But it's precisely this type of interrogation, the one that marked the mentioned transition of pigment protein complexes, if you go from physical chemistry to attract the interest of other communities, and in particular the open system one. So possibly these are the 2 most famous experiments that come from the group of Graham Fleming in Berkeley. There are many other results by other groups. And the initial claim is that the oscillatory features that they were obtaining, typically in cross peaks that would then be allocated to inter-excitonic coherences, they were persisting for a time scale that they consider that was surprisingly long. So let's see what this means. Okay, needless to say, in principle, from the theoretical point of view, one is able to compute what would be the spectral response. In principle, if you have a propagator, right, then it should be possible to compute the 3rd order polarization that is actually the measured quantity. Okay, but more prosaic, and to understand what is the meaning of this long-lived coherences, if you think a much simpler configuration where we have a typical interferometric setup that only concerns 2 time-separated pulses, and imagine a particularly simple scenario where we have a dimeric structure, so just 2 chromophores that coupled will yield to 2 excitonic states with a certain splitting. If the first pulse allows you to build a superposition between the 2 excitonic states, then this superposition will acquire a phase relation during the time in between the 2 pulses, right? That will you subsequently read out. So should the time evolution be unitary, then you will encounter, well, the typical sinusoidal signal that you link to the existence of coherent superpositions. If you do now an experiment actually in a real open system, this theoretical full amplitude oscillation will now have a decay, right? And is the duration of this oscillatory period, right, versus what are the typical oscillatory length lifetime of a ground state, excited state superposition, the one that trigger questions, right? Because a typical value for what will be a ground excited state superposition in a monomer, right, is of the order of 50 femtoseconds, let's say, while the 2-dimensional spectroscopy showed oscillations that were persisting even at 2 picosecond timescales. So, tasks to do. Why do these oscillatory signals persist, oscillatory signals that for the moment very vaguely linked to the presence of coherence in the system. And second, what is the nature of those oscillations? So what is the underpinning dynamics? What is happening in between these electronic and vibrational degrees of freedom? What is happening at the microscopic level, right? That then manifest spectroscopically in this manner. That would be the first question. And the second one will be saying, okay, imagine that we understand the nature of these coherences, and indeed we agree that coherence persists on timescales during which something relevant happens in a biological process. So for instance, a typical timescale from energy transfer from an antenna complex to a reaction center in the mentioned FMO is of the order of a few picoseconds, right? So if there is coherence in that time scale, is it instrumental for something? Is it the case that some process happens with a higher efficiency or even is facilitated at all, like faithful teleportation is facilitated by shared entanglement? So I would argue that the second part of the question is entirely open. While I think we do have some further insight on what are the mechanisms that are involved in the persistence of coherent effects in the spectral response. And moreover, we also have a clearer and clearer picture of what is the associated dynamics. Right. So if you would have heard about this story, right, and have access to the experiments of Fleming and trying to do some theory to see how you can understand that indeed you have a high quantum yield, here quantified by the fact that essentially every photon that is absorbed by the complex will be transferred to the reaction center. You could try to make a division saying my system of interest is actually this chromophoric network that are dipolarly coupled, and I will put all the vibrations as forming part of an environment. An environment that would be responsible for the information from molecular dynamics that is mainly producing local dephases in the chromophores, at least in the relevant time scale. But if you think about rewriting the equations of motion in the excitonic basis, this local dephasing, what is allowing is excitons to relax and energy to be transferred across the complex. Right, so just with this very simple framework, we can see that it's very likely that these complexes operate in a regime where they exploit both coherent and incoherent phenomena in such a way that there is an optimal value of the noise that maximizes the efficiency in the sense of being able to efficiently convey energy in the required time scale. Obviously, this type of noise-assisted transfer is a process that manifests nicely in the laboratory should the right conditions be present. Take into account that the modeling we've done for the moment is not very sophisticated so far. But this is just an illustration of how indeed we observe this type of phenomena in an array of 10 ions in the group of Innsbruck. But it would be quite adventurous to argue that this is really what is going on in this type of photosynthetic complexes if we look at what is the information that we have concerning the spectrum of the fluctuations. So you see that is not really the type of thing we would like to see when we are trying to derive typical Markovian master equations. right? And the issue then would be how we can help further, right? How can we actually argue that we have what we are modeling has anything to do with this type of complexes? In principle, you can try to use the typical strategies we deploy in the presence of structured environments, right? If we have a look at what would be the typical energy gaps we have in our system as a result of the electronic coupling, what we identify quickly is that there can be modes sufficiently narrow. Typically, there could be in most complexes modes that have a width of just of the order of 1 picoseconds, so clearly underdamped vibrations, right? And then we would say, okay, I will just redefine the boundary system environment in such a way that I include within the system whatever is in the vicinity of the excitonic gaps, right? And the residual modes I treat in terms of some residual spectral density. So we know that there are many methods to do that, right? And also with increasing level of accuracy, but the protocol becomes quite questionable very quickly when we start to look at the typical parameters that concern the coupling electron phonon in different photosynthetic complexes, right? So let's see how far we can go, not trying to derive reduced equations of motion, but actually adopting a numerically exact strategy, just not to be ransom of any further approximation. And of course, if we think about doing things numerically exact, then we have to pay the price of reducing the complexity of the chromophoric network. So let me start with the first non-trivial one that would be just dimeric. So in fact, the numbers I will be using in the simulation are motivated by the lowest exciton states of FMO. I start ab initio, writing what would be a Hamiltonian for the whole system concerning both electrons, right? And a local dephasing coupling to a vibrational mode. If I rewrite the total Hamiltonian in the excitonic basis, then the role of the modes becomes quite attractive, let's say, from a quantum optical point of view, in the sense that you see that now I have the possibility of driving transitions in between the excitonic states with a coupling strength that is modulated by these numbers that are actually accessible experimentally, so-called Juan risk factors that characterize the strength of the electron phonon coupling. And part of the, if you want, the intensity of the driving is also the degree of delocalization induced by the electronic coupling. So we exploit here techniques that have been developed in the context of quantum information, mainly in condensed matter, in such a way that can make a unitary transformation for a typical, from a typical star configuration, right, into a semi-infinite chain that can then be modeled numerically using time-dependent renormalization group. The point is that I can now input a spectral density that may have a broad background, right, but also include some sharp features. And if I do that, take into account that here with this type of analysis, I can access the characteristics of the reduced density matrix of the electronic degrees of freedom, something that is not accessible experimentally in a direct manner. But let's say as an exercise, what I can see is what is actually the effect of having these sharp features in my spectral density, and I can clearly see that depending on whether they are absent, like in the dashed line, or whether they are present, the time duration of the oscillatory phase can indeed be enhanced in the presence of this type of underdamped vibrations. More relevantly, even if I do the calculation at room temperature, I seem to have this assisting effect that in a cartoon manner, you can think of having a mechanism built in, provided by the quasi-resonant modes with the excitonic transitions that can repump the coherence that is lost due to the other off-resonant environmental modes. Now, this may sound like a blah blah story, right? Let's say that within that framework, it would still be feasible, the fact that the initial allocation that was done, these are experimental results in the group of Greg Engel, of associating the oscillations to purely electronic degrees of freedom could be tenable. Nevertheless, if we look at what is the configuration, right, and given that even if this oscillatory signal is close to the excitonic splitting, right, can also be instead a very boring story where the oscillations just really come from the ground state dynamics. And simply they have the same frequency, because this would correspond to just a coherence in the ground state between having no phonons and one excitation in the relative motion. If we do actually, and these are already results quite old, 2013, if we look at the spectral response that we can compute, not exactly, so this would be a reduced model that includes the shaft mode as part of the system, what we see is that despite we may have this assistance for the purely electronic coherence to be longer lived, if we look at the spectral response, which is actually what is measured, we see that the origin of the coherence is in fact mixed. So the signatures that we have are consistent with having significant hybridization with this sharp mode. So let's say that already in 2013, the initial bet that the main responsible for the coherent oscillations was of electronic nature, I would think would be disputable. So how things have evolved? Well, this is a recent review that involves, you see a large number of people in the community, with also a large number of people of the community not being involved, confirms, I've tried to underline the most significant part of the abstract. So I read out, extensive research was motivated by the hypothesis that nature used quantum coherences to direct energy transfer. These rest on the interpretation of small oscillations, blah, blah, blah. And they claim is the electronic component, so the inter-exciton coherence, are too short-lived to have any functional significance in photosynthetic energy transfer. And they allocate the largest contribution really to the vibrational component. They finish saying nature, rather than trying to avoid dissipation, exploits it by engineering of exciton bath interaction to create efficient energy flow. So you see that on the one hand, this last sentence leaves us a little bit like in the beginning of the discussion, in the sense that yes, it's indeed quite likely that the interplay of noise and unitary evolution, Hamiltonian coupling underpins the efficiency, yes, it seems that the coherence response is mainly dominated by by vibrational components, right? But the intricacies of how indeed this interface noise coherent dynamics actually happens, I would argue that is still to be debated. And moreover, and here comes the last results I want to discuss, to me, it's still ambiguous determining how relevant the electronic component is. Okay, so how can we get out of this conundrum? Well, we have to see what we can say when we face the full complexity of the environmental fluctuations. So this is a work that started long time ago from our group with Thomas Renga, which is, well, in particular, I think the person that knows more in the world about this water-soluble chlorophyll protein in cauliflower that is also very well studied and even less complex than FMO. So then the issue is that in this recent work, we were able to model what is the dynamics in the presence of the full spectral density. So we are not performing any embedding, and we are considering now the 55 underdamped modes that correspond to intramolecular modes of chlorophyll A, right, together with the broad background together with the low energy part that corresponds mainly to protein vibrations. So we consider water-soluble WSPC as an example of a dimeric system whose excitonic gap falls, you see, in the lower part of the spectrum, so far away from all these high energy tail of underdamped modes. And a very WSCP is probably totally irrelevant, let's say, from the biological point of view. But the second system is key in biology, given that it's the special pair that appears in bacterial reaction center. So the main responsible for actually creating charge separation and then trigger the second phase of photosynthesis. So here the excitonic gap falls full in the high energy part of the spectrum. Right, so let me emphasize again also one issue in case you want to have a look in the literature. In order to make the fitting with the different forms of spectroscopy, right, the numbers that are currently in use for the electronic coupling used embeddings. Embeddings in the sense of first redefining the spectral density. This is typical in the spectroscopy community. They consider our standard spectral density now divided by omega squared. So then essentially we are providing the distribution of the Juan-Rees, the distributions of the coupling. But that can be a little bit dangerous because then the high energy modes, you see, when you perform the embedding of the full red distribution, and you either consider the red curve, right, where you simply ignore any high energy parts, or whether you are a little bit less tough and still smooth it out, this high energy part with these two broad Lorentzians. So we want to do the actual simulation and then compare it with the results that are obtained, typically performing a calculation or a fitting just with the red curves that ignores completely these high energy modes. You can say, well, the couplings of to the high energy modes are very low, yes, but there are very many. Okay, how am I doing with the...
So there is about 10 minutes left.
Okay, perfect. Right, so let us do first the WSCP, because that will provide a nice example of how actually this high energy tail can be the responsible of a large renormalization of the system parameters. So here I have in red the actual spectral density, and you see the embedding in red. There you can say, well, I believe that results are going to be different, but you see that re-expressed in terms of the boundaries, actually the red curve doesn't look like a two-part approximation. Right. Should you proceed, and this is the source of the typically tabulated value for the bare electronic coupling in WSCP. What you would do is to compute the absorption spectra, right, and fit it with the with a green curve that is actually the experimental absorption at 77. Then, from what is the distance in between the two main peaks, corresponding to the higher and lower exciton state, then you conclude that the gap is approximately, and this is a homodimer, so it would be twice the electronic coupling. So then you conclude that the value of the electronic coupling in WSCP is 70 inverse centimeter. Right. We can anticipate that things can actually be different when we take into account all modes in the spectral density. We can compute what would be the vibronic landscape, and then realize that depending on whether the gap in between excitonic states that contain no photons, right, is larger or smaller than the frequencies that are involved in the high energy tail, then there can be multimode mixing that may reduce or may enlarge the excitonic gap. And in fact, one even do a back of the envelope calculation using perturbation theory to see that there can be a strong renormalization effects. So let's see whether that's the case. So one of the things that hindered the simulation of the whole spectral density, of the whole dynamics and the whole spectral response with a complete spectral density was the fact that if we wanted results at high T, the method I told you before is not very efficient. So we rely now in more powerful results, on the one hand, showing the unitary equivalence between different configurations, and especially relevant for the numerics that I'm going to show you, is a work in 2019 where we can unitarily map out the temperature dependence that in our simulations enter as the initial distribution of the vibrations, right, is now mapped up in terms of a temperature-dependent spectral density. This provides a significant simplification to be able to operate efficiently at a higher T. Okay, so this is exact absorption spectra, also using two different methods. We use also the hierarchical equations of motion that Walter was mentioned in his talk before. And if you fix your attention in the panel C, we can actually check what is the effect as we include N modes 40 or 55, and how do we need to make a full consideration of all of them in order to accurately reproduce what is the measured absorption spectra? Now, this has very relevant consequences, because if I look at what is the typical value considered for the electronic coupling determined by a fit in the spectroscopy with a smooth spectral density, I realize that if I use that value now with a full spectral density, my prediction is considerably far off. Okay, so one effect that we have from the presence for the full consideration of the whole spectral density is that there will be complexes where there is a renormalization of the system parameters. In particular, the electronic coupling could be considerably underestimated by means of doing fittings with the smoothed out forms of the spectral density. Okay? So in fact, in the particular case of WSCP, right, in order to reproduce absorption with a full spectral density, the value of the coupling has to be roughly double. And moreover, and this brings us to the discussion of the origin of the coherences that we are observing in the spectral response. If we consider now the biologically relevant complex with this special pair in a bacterial reaction center, which corresponds now to a situation where the gap is actually falling within the high energy part, then we see that there are clear difference in the value of the excitonic coherence, depending on whether I make the calculations concerning the spectral density that includes the full complexity, or whether I'm performing smoothing out over the high energy part. In particular, we can, well, now we can, but with different techniques, but let's say in the paper I just mentioned, published last month, the two-dimensional calculations that we do are actually based upon a reduced model that doesn't include all the 55, but actually 6 of the high energy modes. But the point is that if we look to the oscillatory signals, they do vary. And if I have a look at the form of the responsibles for this mixed coherence, that will now be coherence between dress states, between vibronic states, I do have contributions that come from purely excitonic, right, to indeed others that would be more dominated by vibrational degrees of freedom. But going to the diagrams itself, so if I look at what would be a typical signal that goes almost up to a 1 picosecond, we do have contributions of purely electronic origin, right, together with others that are of vibrational origin, that when we make a Fourier transform, do correspond to a splitting that is now the renormalized excitonic separation. So what is the take-home message of all these? Well, we are in the presence of systems where, given that full tomography is, or process tomography is really not accessible, it's not at all trivial to reconcile within a single model both the spectral response, right, and also the underpinning dynamics. So this is what so far has prevented the entirely unambiguous interpretation of the experimental results as far as the microscopics is concerned. So This is a cartoon that I created almost 10 years ago. So let's say that this picture of indeed a nice interplay between noise and coherent dynamics remains, right? But the intricacies of how it's actually happening, I would say that there are still many issues to be uncovered. And definitely one important step forward would be having a new generation of experiments that can actually be guided now by the possibility to doing numerically exact calculations of the spectral response, also multidimensional, with the full spectral density. And then singling out, right, the specific features that we can link to features of the dynamics. That you can say, well, maybe that's not that relevant. Yeah, let's say which percentage of the coherence is actually of electronic origin, which one is vibrational. But I think especially if we think of then using this analysis maybe as an input for the development of nanofabricated devices, I think it's important to really fully understand what is pulsating underneath the spectral response. The big question, is it actually relevant that whatever its nature, the coherence is actually deployed for biological function? I think this is open, and I think one way forward is really discriminating families of theories that can achieve this efficiency or this another. So I always put the example of the Bell inequalities in the sense that we can then discriminate definitely theories that are not able to account for the observations from those that can be. And there will be many theories that can account for linear spectroscopy, but then some of them will fail when they have to also reproduce the multidimensional spectroscopy, right? And then we can also characterize in terms of some reasonable measures of efficiency, right? For that, a thermodynamical picture is nice. What is the maximum power that a given complex can develop if thought as an operating between two reservoirs, very much like a thermal machine? Then we could progress forward towards really understanding whether indeed there are some tasks of relevance in biology that can be assisted unambiguously by coherence. This has taken several years. So this is people that have been in the group collaborating, and in red, including our late Felipe Caicedo, the people that have been involved in most of the work I told you about today. Many thanks for your attention.
Thank you very much, Susana, for this very exciting talk. So we do have time for maybe one or two fast questions.
Stop sharing.
So let me ask very fast and simple question while the other people are still giving. So you mentioned this next generation of experiments into artificial complexes, right? So what kind of physical devices these are?
Right. I would say that with artificial complexes, there are experiments that really you can reconcile with a dynamical model, right? So let's say the mechanisms that are at play, there you can make a correspondence. What I think it would be particularly relevant is having, again, experiments done with natural complexes, so even using FMO again, but now will be more targeted in the sense of the advantage and at the same time the disadvantage of the multidimensional response is that it contains a lot of information superimposed, right? So now it would be good that experiments are guided in the sense of now identifying selected parts of the frequency ranges, right, that can then be confronted with an associated dynamics.
Right. So making the connection between the...
Exactly.
Right.
Can I make a question? From Palermo University. Just perhaps I lost something. What about the temperature? You in your model you fix the temperature. And second question connected eventually. Perhaps I lost something. Did you consider thermal fluctuation or noise environment? This is the question.
Yeah. So the simulation is actually the last ones I told you about. They are exact. Right? So you are accounting for the full noise if you want. You want to call the system your electronic degrees of freedom, right? They are feeling all the fluctuations that are due to their own intramolecular modes and all the protein motion, right? And the full spectrum of those fluctuations are considered. And indeed, the numerical results I showed you range from 77 to actually room temperature.
Okay.
And this is another interesting thing. Let's say that your model needs to be robust to all available spectroscopy. So you have to make accurate predictions on low T, you might have to make predictions at room temperature. Of course, for anything coming out that is of interest in biology, it has to be sufficiently robust to survive at room temperature.
Robust, even if you change the temperature.
Yeah, your model should be robust to changes of temperature, but let's say that whatever you found that you think is interesting, if it's only visible very, very cold, then, well, maybe you can exploit it for some tasks, but you cannot claim that it will be relevant in biology.
Okay.
So there we need coherence robust
in
hot environments.
Thank you very much. Thank you.